Okay, so for this video, we're gonna just do a basic review of the nervous system. Now, as I've said about other topics in this class, there is an expectation that you've already seen some of this. So I'm not gonna go incredibly in depth into parts of the nervous system, central, peripheral, um, sympathetic, parasympathetic. Like, I'm not gonna go really detailed in how a neuron depolarizes. I'm assuming you know or have had that in biology. For some of you, anatomy and physiology. Others of you, human structure and function with me. And so, if you haven't had that, if it's been a while, if you just just don't want to remember that, in on Canvas there are some links to videos there that will hopefully kind of catch up to speed on some of those topics as so you kind of are where everyone else is, hopefully, as far as the class goes. So in this part of the nervous system, we're just going to do a really quick overview. We're then going to get into motor neurons, how they function, and then we're going to spend a lot more time on more nervous system with muscle, referring to proprioceptors, chemoreceptors, and then kind of finish with parasympathetic versus sympathetic and how that has a role on not just muscle function but other processes in the body. So this is just a basic overview. This is just a basic overview of the nervous system. We are, again, we're not going to go terribly in depth in this. And the reality is we're going to spend most of our time here in the peripheral nervous system, specifically in the sympathetic and parasympathetic part. So we will talk about the central nervous system a little bit, brain and spinal cord. We will talk about involuntary contractions, a very small amount, but the vast majority is going to be here in the sympathetic and parasympathetic nervous system. So again, when we talk about a neuron, basic structure here that again I hope you've had before. There are neurons in all mammals, whether you are a ferret or human, even small worms have neurons. You have billions upon billions of these in your body. And though they all have somewhat different look to them, some can be as long as a meter, some can be microscopic, quite literally micrometers long. They all have very similar structures in them. So they are going to have a cell body, and this is where the nucleus is going to be. There'll be dendrites that extend out from here. This is where all the typical parts of a cell we'd expect to be: a nucleus, endoplasmic reticulum, some mitochondria, what we typically think of when we think of a normal animal cell. Then we're also going to have our axon hillock, then our axon. Our axon is what the action potential will travel down, and the message will travel down. And that'll go down to end terminals or synapse terminals, motor end terminals, whatever you want to call those. And that is going to then create a synapse with another cell, another neuron, another something to continue on sending that message. We've also talked about previously, at least in other classes, this idea of a resting membrane potential. The idea that these neurons have a charge, they have a voltage. And so quite literally, if we were to put a voltage meter inside a neuron, in humans, it would be somewhere in the ballpark of about negative 60 to negative 75 millivolts. So our inside of our neurons are more negative, we could say, than on the outside. And this charge is because of ions. And if this is sort of an example here, there are a lot of ions we could talk about here. There's proteins here that are adding to this. But the two big ones we talk about are sodium and potassium. If you've had me for other classes, we talk about these salty, sodium, banana, salty banana. The idea that there are channels here that are going to play a role in how these ions, whether they stay inside or outside the cell, whether they stay there. And at normal rest, when a cell has its rest, that potential will stay at negative 65, negative 70, whatever that membrane potential is. And it will take some stimulus to cause that to change. However, that's necessary to send a message down a neuron. When a message travels down a neuron, it travels down a neuron because that neuron depolarizes. In other words, it'll change that potential, it'll change that voltage to something much more positive. and that's what causes a message to travel down a neuron. And so just understanding that there are channels here that are associated with keeping this potential normal. We call them sodium-potassium pumps. Those sodium-potassium pumps require ATP, and so not surprisingly, keeping this membrane potential normal requires energy. And we've obviously talked about ATP, how we form that. Your nervous system, by the way, consumes a lot of ATP. Upwards of the ballpark of about a quarter of the ATP you use is because of neurons and them continuing to function. And so the key role here, and obviously in using ATP, using kilocalories during the day. And so these are what some of these channels look like. Again, there's a lot of ions here. Chloride is another one. Proteins, by the way, are a large reason why there tends to be a negative charge inside the neuron. They tend to have a negative charge. And there are these channels, these protein channels, that keep sodium in, sodium out, potassium in, and keep those concentrations normal. However, what occurs is when a message is going to travel down a neuron, it causes what's called an action potential. And that is when there is some stimulus, and this can be numerous things, but if we want to talk about skeletal muscle, if you want to move your finger, a stimulus would be something in the central nervous system attaching to a neuron or creating a synapse with a neuron, releasing enough neurotransmitter to cause that neuron to depolarize, basically to tell a muscle cell, hey, we'd like you to contract. And so when that occurs, really briefly, you have sodium channels that are going to open up. Sodium comes entering through the cell through those channels. As that does, sodium has a positive charge, so that sodium positive charge causes the membrane potential to increase, become more positive. We call that depolarization. In response to that, those channels close, potassium channels open, and when these potassium channels open, sorry about that, my cursor's not working. When those potassium channels open, we call that repolarization. Potassium is going to enter back into the cell. That is going to cause this then to become negative. It actually becomes over negative for a very, very brief time. And then we go back to our resting membrane potential. And there is all action potentials are the same size. So there is no like partial action potential. There is not a small one or a big one. We call that an all or none principle. The idea that this will occur totally or it will not occur at all. And these are sort of the steps here. I'm not going to go through these because I know you've had these elsewhere as far as these channels opening, depolarization, repolarization, and then returning back to our resting state. There's some videos here. Again, you can take a look at, because again, we're assuming you know you've had that in other class environments. And this is sort of a picture of what this looks like as these channels open, moving down our neuron. And if you've had this before, you know we're ignoring myelin sheath, we're ignoring some other things that assist in this depolarization. But here we're having our depolarization as the sodium channels open up, and then repolarization as these return back to normal. Okay. This is where I want to slow this down a little bit because there are types of nervous tissue throughout the body. And, we focused, at least recently, on motor neurons sending messages to muscle cells, getting them to contract, getting the muscle to contract. However, there are other types of, we'll just call nervous tissue that exist. And we're going to call these receptors or sensory receptors. And these will also depolarize. They also send messages, afferent messages, to the central nervous system, allowing us to understand different qualities or different things about the environment. For example, there are mechanoreceptors. Mechanoreceptors exist. We'll talk about those that sense actual force. This might be for tension produced in a muscle. It might be stretch. And that's going to send messages to the brain to say, hey, muscle is contracting. Something is going on. There are other receptors, thermoreceptors, which are sensitive to temperature. And I think we can all understand those. Nociceptors, which are sensitive to pain. Chemoreceptors, very important, are sensitive to chemical changes in pH, things like that. And all of these are going to send sensory input, afferent signals, to the central nervous system, which then will cause some efferent response. This could be movement of muscle. This could be, a reaction to a temperature. It might cause shivering if the temperature is very cold, an involuntary muscle contraction. This may cause a change in gait if there's pain. This may cause an increase in breathing. If pH is dropping, we need to get rid of CO2, which is a byproduct of those hydrogen ions being produced. Photoreceptors, it might be altering the lens shape, maybe closing our eyes, using the muscles around our eyes to do that, or move eyes. So there will be responses to these various types of input. Now we're going to hang out on these proprioceptors because they're a little bit more about physical motion. They're more related to skeletal muscle, which we're talking about. So I want to hang out in these a little bit more, and that's where we're going to go to in our next video.